Now, metabolic reactions, we will be running many of them, but oftentimes these metabolic reactions are going to require catalyst. Well, the catalysts that we tend to have inside the cell are organic catalysts. Catalysts in general are going to be chemicals that increase the likelihood of a reaction taking place. So in other words, they are going to help the reaction occur without themselves being actually used up or consumed in that particular reaction. So as a helper, they would be able to repeatedly help that reaction again and again and again because they themselves are not being used up. The organic catalysts that we will be primarily using are called enzymes. And those enzymes are mostly made up of proteins. And if we look at the way that our reactions will be drawn when we're using these enzymes, so we will start with a reactant, which we're now going to call a substrate. And that substrate is going to get ultimately converted into a product. The enzyme, since it is not being used up, it does get written there in the reaction right above the arrow, again just indicating to us that it is being used. It's there at the beginning of the reaction, it's also there at the end of the reaction. So we don't really consider it a reactant or a product in this case. There are 6 basic enzyme categories, and here they are. The first one is a hydrolase. Hydrolases are going to add water to molecules, which will ultimately result in their breakdown. So they would be breaking substances up. Isomerases are going to rearrange atoms within a molecule. So when they rearrange those atoms, we call the rearranged atoms isomers. And so isomerase is essentially creating isomers. Ligases or polymerases are joining or connecting molecules together. Lyases are splitting large molecules, but the difference between this one and the first one, the hydrolases that we listed, is that the lyases themselves are not actually going to use water to make that happen. Oxidoreductases participate in the redox reactions. So these would be completing an oxidation portion and then also a reduction portion. The last one here, transferases, are going to transfer functional groups. And this could be transfer of an amino group, transfer of a hydroxyl group. There's lots of different functional groups that could be transferred here. Now, one thing you might have noticed about all of these different enzymes that we've listed is that they do tend to all end in the suffix ase. So if you do see a word that has ase on the end of it, that's a good clue that you're looking at an enzyme, and that enzyme will be performing some type of a reaction inside of our cells. So how do the enzymes work? Well, enzymes, they are mostly made of protein. Most of the time they're made of protein, but we do have some cases where they do happen to be made of RNA as well. And so the blue that we see in this picture here represents the enzyme itself. And enzymes sometimes, even though they are helping a reaction, they need a helper for themselves. So the helper components are called cofactors or coenzymes. The difference between them is that a coenzyme is organic, which means it's probably going to be bigger. This might be something like a vitamin, and we can see how that fits right in there with the enzyme. And then the organic cofactors are usually quite small. This might be just an ion, maybe like a zinc ion or a magnesium ion. So if the enzyme requires it, it is going to need its cofactor and also its coenzyme to be there present with it to actually get it to a working functional enzyme. When all of it is collected together, we do call it the holoenzyme. Once all of this is kind of put together as well, we're going to have a special place on our enzyme that's called the active site. And the active site is going to be specifically where the substrate binds. So whatever that substrate happens to be, it's going to fit perfectly into that active site, which means that our enzymes are going to be very specific molecules. They're definitely not a one-size-fits-all situation. So if we look at the fit of the substrate into the active site of the enzyme, we see that right here. Once the substrate fits into the enzyme's active site, we get this complex, which we call the enzyme substrate complex. And at that moment in time is when we actually see the enzyme performing its job, which is helping the reaction to take place in a quicker fashion. So making it more likely that the reaction is actually going to occur. Enzymes specifically are going to lower the activation energy of the reaction. And the activation energy is the energy amount that we need to get this reaction going. So we think of it like a hill. If you look at this diagram right here, the reaction without the enzyme being present has a nice tall hill. And so we're talking about starting here and climbing all the way to the top of the hill. So if we had to make this climb, that would be tougher than making this climb down here and only going to the top of the small hill. So if we only had to go to the top of the small hill, we can get there faster. It's going to be more likely that we do actually get to the top of the hill. So enzymes are basically going to take the hill from being up here to being much lower. And so that is what they do. They lower that activation energy, which in other words, another way to define this is it is the height of the hill. And again, we're making it lower. If we lower it, then we're going to be getting this reaction happening much more often. Okay, so here's a look at an enzymatic reaction, so a reaction that does involve an enzyme. The enzyme is the blue structure, and notice how you have a molecule that's going to fit right into that active site. So the substrate comes in and fits into the active site. We then get the enzyme substrate complex, and this moment in time right here is when the activation energy, which we call the EA, that is when the activation energy is lowered. So that is when we're going to see that hill get shorter, and at that point we're going to be able to very quickly climb the hill, and notice that we do get our product formed, which in this case is actually splitting the actual molecule in half. So splitting that fructose bisphosphate that we see here. How the enzymes actually work, how they actually lower that activation energy, we don't know everything about that. We don't know exactly how it happens, but some of the ideas behind it is that maybe the enzyme is kind of twisting those bonds so that they're strained and they want to break, or they're more likely to break if we do strain them a little bit. Okay, but something about being in the active site there is going to lower the activation energy. Besides the enzyme itself, there are other factors that are going to influence reaction rates. One of the most common things that we think of that influences a reaction rate is temperature. In general, as we increase the temperature, we see reaction rates go up. So increasing temperature can be a good thing, except for the fact that if we increase the temperature too much, it can be a problem for the enzyme structure. Enzymes are proteins, which means they're very susceptible to temperature changes, which could cause denaturation. And denaturing is basically taking that perfectly folded enzyme and unfolding it. And if we were to unfold it, we would be unfolding the active site at the same time, which would cause the enzyme to become non-functional. So extreme temperatures cause the enzyme to fall apart. And that's what we see happening in this diagram down here. The protein was folded, we changed the temperature from the ideal temperature, and now we have a protein that is denatured. It's not going to work. It's not going to be able to perform the same reaction anymore. If we look at this chart right here, which is showing for a particular human enzyme, what temperatures does it work best at, notice that the best temperature for this particular enzyme happens to be what our human body temperature is. And that would be right at 37 degrees Celsius. Our enzymes ideally want to be at 37 degrees Celsius. And that is one of the reasons why our body temperature has to stay right around 37 degrees, because that's going to ensure that those enzymes are properly folded and able to perform that particular reaction. Now, if we vary too much on the high side, we start to see a drastic drop-off in our enzymes' ability to work and function. If we go to the cooler side, we can see that we're getting a steady decrease on that side as well. And if we get too far away from 37 degrees, then the enzymes aren't going to function at all, because at that point they would be unfolded. pH also has big impacts on how that protein is actually folded. So changes in the pH can also drastically affect the ability of an enzyme to function. Our enzymes are also designed to work at specific pHs, and those pHs would need to be maintained. If we had stomach enzymes need to be able to work at very acidic pHs, because that's what the stomach environment is like. If we're talking about intestinal enzymes, they have their own ideal pHs, and the intestines is more of a basic environment, so those are going to be enzymes that are designed to work at the type of pHs that we would find in the intestines. Increases in substrate concentration can also increase reaction rates. This is up to only a certain extent. This is up to what we call the saturation point. So if a cell needs to run a reaction at a much more rapid pace, it will increase the substrate concentration. The enzyme will just keep grabbing hold of that substrate in its active site, lowering the hill, and we get product. But again, notice that after we increase at a certain point, the saturation point, at that point the enzyme is basically working as fast as it possibly can, and we're not going to be able to increase the reaction rate or increase the production of product anymore beyond that particular point. Cells also control reaction rates in a negative way, and when they do this, they're actually lowering the ability of the enzyme to decrease the activation energy. The molecules that are going to be lowering the enzyme's activity are known as inhibitors. These inhibitors specifically bind to the enzyme and alter the enzyme's ability to function. They do work in a couple different ways. One type of inhibitor is called a competitive inhibitor, and the competitive inhibitors actually bind to the active site and block the binding of the substrate. So they compete for the active site. Sometimes these are non-reversible. They bind in there, and if they do not come out of the active site ever, then that means that that particular enzyme is not going to be able to function anymore. Well, a lot of times we do want to be able to reuse the enzymes. We would want them later to be able to actually run that same reaction again. So reversible competitive inhibitors are very useful. With a reversible competitive inhibitor, the cell can actually outcompete and get that inhibitor out of the active site. One way that it does that is it increases the substrate concentration, and if it increases the substrate concentration, then the substrate has a better chance of getting into the active site, and when it gets into the active site, we will have this particular reaction happening again. So this substrate molecule is going to be bound by an enzyme, and this enzyme is going to convert it into an end product. And we can see that happening down the side of this diagram. Well, if the end product becomes abundant, so if it becomes too much, that would indicate that the cell is not using that up. So it's building up, stockpiling, which means that the cell probably doesn't need to continue to build this anymore and make this anymore because we already have too much of it. So what happens is the end product itself acts as an inhibitor right here on this enzyme that functions earlier in this particular pathway. So it binds to that enzyme and it shuts it down. So at that moment in time, that enzyme is no longer going to help to produce the end product. Now, if the end product starts to get used up, so our stockpile is dwindling and going down, 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 eventually we will use this one up too. And when we use that one up too, notice that the enzyme now starts functioning again. So now we're going to start to make end product. So what this means is that as long as we are using up whatever we're making, we're using it up, then this whole pathway is going to continue to churn that particular product out. But as soon as this starts to build up, we turn it off. And then we wait until this gets used up, and then we can start making again. So In this case, if too much of a product builds up, then it shuts down its own synthesis. So that's what we call feedback inhibition. It's just a really good example of how the cell uses inhibitors to balance supply and demand.